Hello guys, welcome to my channel. So as you know, we are studying about the protein. We studied about the fibrous protein and the globular protein. So now we'll see the protein quaternary structure a, just a little bit in detail, and then we'll do the protein denaturation and folding. So just to know that the protein quaternary structure range from simple dimers to the large complexes. So write that down. Protein quaternary. Protein quaternary structures range from simple dimers to the large complexes, right? To large complexes. Okay, so what does that mean? So that means that many proteins have multiple polypeptide subunits, right? As we know that it can range from 2 to 100, and the association of polypeptide chains can serve a variety of functions. For example, many multi-subunits protein have a regulatory roles, correct? So the binding of the small molecules may affect the interaction between the subunits. So for example, if the protein is serving as a regulatory role, and if the protein needs to bind to a small molecule, but it has a multi-subunit, so what happens sometime when the small molecule binds, so it can affect the interaction between the subunits, so causing large changes in the protein's activity. So causing large changes in the protein's activity in response to the small changes in the concentration of that substrate or the regulatory molecules that has attached. So there is other case as well, like in other case, the separate subunits take on a separate but related functions such as catalysis and regulation. So some associations such as fibrous protein that we saw further, so that's one of the example, and the coat proteins of viruses serve primarily structural role. So they just was the primary role of that protein, that kind of protein is just the structural role, right? And there are some very large proteins that assemblies are the site of complex and multi-steps reaction. So for example, each ribosome, the site of protein synthesis, incorporate dozens of protein subunits along with the RNA molecule. Right? So the multi-subunit protein is also referred to as a multimer. So it's referred to as a multimer. Right? So what is a multimer? A multimer with just a few subunits is often called oligomer. So if a multimer has non-identical subunits, the overall structure of the protein can be asymmetric and quite complicated. So if it's identical, then it's okay, but if it's non-identical, then the overall structure of protein can be very asymmetric and complicated. However, most multimers have identical subunits or repeating groups of non-identical subunits, usually in the symmetric arrangement, right? So the repeating structural unit in such a multiple protein, whether a single subunit or groups of subunits, it is called protomer. So what is protomer? The repeating structural unit in such a multimeric protein where a single subunit or a group of subunit is called protomer. So the first oligomeric protein to have its three-dimensional structure determined was hemoglobin, which contains four polypeptide chains and four heme prostatic groups, in which iron atoms are in the ferrous state, right? The protein portion, the globin, that consists of two alpha chain and two beta chain. So the alpha chain and the beta chain like sometimes confuses students as well. So just in a practice, so the Greek letters alpha and beta are often used to distinguish two different kinds of subunits within a multi-subunit protein, regardless of what kind of secondary structure may predominate in the subunit. So because hemoglobin is 4 times as large as myoglobin, much more time and effort were required to solve a three-dimensional structure by X-ray analysis. So the subunits of hemoglobin are arranged in symmetric pairs, each having one alpha and one beta subunit. So hemoglobin can therefore be described as either as a tetramer or as a dimer of alpha beta protomers. Right? So this is just a description like that. And also there is like some kind of proteins, or there are protein segments are intrinsically disordered, that means lacking definable three-dimensional structure. So these proteins have distinctive amino acid composition that allow a more flexible structure. And some of these disordered proteins function, what's the function of that is as a structural components or the scavengers, so that other can interact with many different protein partners, serving as a versatile inhibitors or certain central components of protein interaction networks. All right, so now we'll study about the protein denaturation and folding. So just remove this. Foldings. All right. So as we know that the native protein conformation, and if you remember what is native proteins, the protein that are in the natural state, that with the intact structure, right? That is not altered by any heat or enzymes or any chemicals, right? So the native protein, the most protein, what's native protein? The protein must maintain the conformational flexibility to function, right? And all the native protein conformation is only marginally stable. So the continual maintenance of the active set of cellular protein required under a given set of condition is called proteostasis. So what is proteostasis? So there is a term called proteostasis. And so what is proteostasis? The right, so the continual maintenance of an active maintenance of the active set of, right, the active set of the cellular protein, right, cellular proteins required under the given set of conditions, that is called proteostasis, right? All right, so the cellular proteostasis requires the coordinating folding of pathways for protein synthesis and folding. So the refolding of protein that have been irreversibly unfolded or are no longer needed. So the cellular proteostasis. So what is cellular proteostasis? So that means in cell the proteins. So the proteins that are newly created, they need to be folded. Some of the proteins that are unfolded, so they need to be folded back. If there is like proteins that are no longer in use, they should be sequestrated and degraded and done, and degradation to be done, right? So this all are the cellular proteostasis.